My name is Dr. Rhiannon Werder. I'm a team leader at Murdoch Children's Research Institute, where I lead research using stem cell models to investigate lung disease. I've always been inspired to learn as the daughter of two school teachers, and then I was really fortunate in high school to have a chemistry teacher that inspired me to go down a scientific career path. My undergraduate degree was in biomedical science, but it wasn't really until I did my honours year that I got a taste of what real science was actually like, and I absolutely loved being in the lab and discovering new things every day. After my honours year, I then immediately embarked on a PhD, and this is where my interest in respiratory disease really started to hone in. My PhD was looking at why early life respiratory infections predispose children towards developing asthma. During my PhD, I received a scholarship to undertake training at Duke University, and this was a really pivotal moment for me because I realized how much I would want to do a postdoctoral fellowship in the United States. So by the end of my PhD, I was really interested in how the lung epithelium goes awry and causes many respiratory diseases to occur. And so I sought out labs that could help me to answer this question in the United States. With the help of a C.J. Martin Fellowship, I undertook postdoctoral studies at the Center for Regenerative Medicine at Boston University. And this, again, was a real turning point for me because it was where I first learned how to use induced pluripotent stem cells to study lung disease. I was also in Boston during the COVID-19 pandemic, and this was a time that therefore allowed me to combine two major interests, one being stem cell biology and the other being infectious disease. So at the end of my time in Boston, I then returned to Australia, where I've now been at the Murdoch Children's Research Institute for 2 years, where I've established induced pluripotent stem cell models to study lung disease. My team and I research a whole spectrum of respiratory disease, from the very common, like the common cold, all the way through to rare lung diseases that affect children. And the reason that we're doing this is that for many respiratory diseases, we lack effective treatments. Part of the problem in developing new therapeutics for respiratory disease has been a lack of models that we can study in the lab to really understand how the human lung operates. This has then led to a slow discovery of new therapeutics. And so the way that my team tackles this is using stem cell models of the lung. And the stem cells that we use are called induced pluripotent stem cells. And these are cells that we can reprogram or reset from blood cells or skin cells from any individual. We can then turn these stem cells into any of the cells that live within our lungs. So that can be the epithelial cells that line our respiratory tract, the endothelial cells that line the blood vessels in our lungs, or the macrophages, for instance, that constantly surveil our lungs for invaders. We're then using these models to study respiratory infections with a real focus on respiratory infections in people with preexisting chronic lung disease. Another aspect that we're looking at is how the lung repairs itself after injury. And this is a particularly important question to answer right now because in the wake of the COVID-19 pandemic, we're seeing many cases of long COVID. And so what we're trying to understand is what balances good versus bad repair after an injury, and can we instead push the lung towards beneficial repair to prevent long-term consequences like pulmonary fibrosis, and ultimately find new opportunities where we could intervene therapeutically. So where we are currently is we've created much more complex models of the human lung, incorporating many various cell types that we've created from stem cells, from the epithelial cells through to macrophages, endothelial cells, and fibroblasts. So for the next 5 years, what our plan is to investigate a whole range of respiratory infections, from rhinovirus, which is the cause of the common cold, SARS-CoV-2, which is a cause of long COVID now, as well as rare but really debilitating bacterial infections like Group A streptococcus and strep pneumoniae. My research is funded by grants from the Australian government, in particular the MRFF, as well as a large global consortium called Renew, which is funded by the Novo Nordisk Foundation, as well as philanthropic organisations like the Stafford Fox Foundation for Medical Research and the Lou Carty Trust. My passion really comes from discovery science. I love the everyday discoveries that happen in the lab, whether they're big or small. I'm also really inspired by the patients and families who we work with on our quest to find new therapeutics for respiratory disease. And I'm also really inspired by my team who I work with every day. Outside of the lab, I really enjoy traveling. I love playing music on the piano, as well as playing golf with my husband. I like enjoying time with my dog, and I also enjoy cooking, which in some respects is a little bit like working in the lab every day following protocols.